Okay, so it's my pleasure to introduce Lev Litichevskiy. He's an associate computational biologist at the Broad Institute, and he is a member of both the Link Center for Transcriptomics and the Proteomic Characterization Center for Signaling Epigenetics. So Lev, please tell us about using sentinel profiles for drug characterization.
Thanks, Jake. So Jake asked me to give this lecture to highlight some of the ways in which all the data that we've talked about producing and processing so far can actually be used to learn something about biology. So I'm going to cover that. But there are two other things that I was hoping to get to in this lecture, which are, one, to walk through post the normalization and processing that you've already discussed. We have some more computational steps, and I wanted to walk through our motivations for those steps and kind of explain what we do and why. So that's going to involve a bunch of topics you've already seen before, but I thought we could slow down and kind of go into depth on a few of those. The last thing I wanted to do is Morpheus, which you've all heard about, I think is a really powerful tool. I use it every day in my work. So I was hoping to do a tutorial with everyone to kind of help you get warmed up to it, because I think it's something you might be able to use in your own work. So hopefully we'll get to that as well. So where we have left off so far is with making profiles. So profiles are median normalized log 2-fold change matrices of data that we often view in heat maps like the one on the right. So in the rows here, we have the 96 P100 analytes, and the columns are the samples. In this case, I'm showing 18 samples of Vemurafenib in 6 cell lines. And this is part of a much bigger matrix that was a big data set that we made. 90 drugs in 6 cell lines profiled with P100, and actually 2 other assays, which I'll hope to get to later, but for our purposes, we're just looking at the P100 data. So it's 540 perturbations altogether, and this is just one part of it. So the slide is sort of to motivate an idea I think you're all familiar with, which is that profiles can be subtle. So these 18 profiles, there's clearly some differences at the analyte level for some of the profiles. Some things really stand out, but it might not be obvious that one of these cell lines should have very different behavior than the other cell lines when treated with Vemurafenib. So to kind of step back and cover why we know that is the case, we have a little pathway diagram here of a receptor tyrosine kinase embedded in a cell membrane. So this kinase phosphorylates a protein called RAS, which then acts on BRAF, which then acts on MEK and ERK, and that downstream leads to proliferation, so cell growth. Now, there's also a version of BRAF where it gets valine at the 600th position mutated, and it becomes either a glutamine or a lysine. And of the 6 cell lines that we have, A375 is the only one that has BRAF mutated, has a V600E mutation. And Vemurafenib is a drug that is supposed to selectively bind the mutant form of that protein. So in theory, it shouldn't act unless there's that mutated BRAF protein around. And when it is present, then it binds it. And to explain the mutated form, so when BRAF gets mutated, it's constitutively active. So it's just constantly phosphorylating MEK and ERK and leading to widespread proliferation. But Vemurafenib is supposed to stop that growth. So the idea is that Vemurafenib should do something very different in A375 compared to the other cell lines, because A375 is the only cell line with that particular mutation. Right? Okay. So if we try to parse out, well, is A375 somehow different than the other cell lines, we could start by looking at individual analytes, perhaps the really brightest ones. And I probably don't have to convince this audience that that is not a very effective procedure, especially with the P100, as you've seen, being chosen kind of randomly out of a huge set of phosphosites that may or may not mean something. So the first site, we actually do find something meaningful. Uniprot, if you look in Uniprot, there's citation, evidence that this site might be phosphorylated by MAPK and ERK. So that's related to the Vemurafenib pathway, so something correct is going on there. If we look at another analyte, nothing. And if we look at the third one, there is information about it in Phosphocyte Plus, but it's a bunch of diseases, a bunch of cell models, and it tells you that one drug might affect it, but that drug I know affects a whole bunch of other phosphocytes just from combing through a lot of Phosphocyte Plus data. So, okay, no big surprise, this isn't the most effective way to look at these data. So we say we should consider the whole profile. But quick step back on what is this profile that we keep calling. A profile is a vector. It's just a vector in high-dimensional space. So if we only measured one analyte, it would just be the vector pointing to this point here. So we measured a value of A for analyte X when we treated with the drug V. If we had two analytes, it would be a value of A for this analyte, a value of B for this other analyte, and now the vector is towards V in this two-dimensional space. In 96 dimensions, we can't visualize it, but again, it's just a point out in some big space. And all that's to say that now that we are thinking about profiles as vectors, the question is, how can we compare whole vectors to each other? So the simplest idea, well, here are a few examples. The first and kind of simplest idea is Euclidean distance. So you have two vectors in high-dimensional space. Just calculate the distance between them. But there's kind of a motivating story as to why we think that's not effective for comparing our profiles. So imagine you have one very long vector pointing in one direction and another vector much shorter in the same direction. You can think about those two vectors perhaps as the same drug but at two different doses. So the higher dose just causes bigger changes in each of the phosphosites than the lower one. According to Euclidean distance, that could be quite a big difference if the one vector is much longer than the other. But we want a metric that somehow tells us that something similar is going on there if we're treating with the same drug. It's just that the dose is different. So we want to find, we're kind of looking at the angles of the vectors. Are the vectors pointing in the same direction? So that's how we get to correlation, which is really familiar to everyone, but kind of I wanted to build up to why we settled on correlation. And Pearson correlation is kind of the standard option. If these two vectors were centered, so if their means were right at the origin, then Pearson correlation is just exactly the angle between those two vectors. So it's exactly what we're looking for. But Spearman correlation is just slightly different in that you first convert the values to ranks. So it doesn't matter so much if the very highest phosphosite value is 10 or 20. If it was the highest one, it gets assigned a rank of 1. And as far as Spearman correlation is concerned, that's exactly the same. So I want to spend a little time going deeper into the differences between Pearson and Spearman because those are kind of, in our eyes, two very plausible similarity metrics, and I want to justify a little bit why we chose Spearman. Any questions so far? Is this doing fine? Okay, so this is straight from Wikipedia, explaining some toy examples of where they're different and where they're not different. So you've got, say, some treatment here, some treatment here. Each point here is the value reported for a phosphosite. And Spearman says this is a perfect correlation. The value that's the lowest in one sample is also the lowest in the other one. The value that's the highest is the highest. Perfect correlation. Pearson's also high, but not as high. In the middle, the correlations are about the same, but the place where they really kind of diverge is when you have one sample that has some outlier values, but there's still some of the highest values, right? So these points here are some of the highest values in both cases. It's just that the absolute value of them is much different in this sample than in the other one. So Spearman there is a little higher. And that's kind of our motivation as to why we think we prefer using Spearman, is to be able to tolerate those outlier values a little bit better. But this is toy data, so if we look at actual data, bringing back the Vemurafenib samples, if you correlate two of these vectors, right, these profiles, and these are two replicates in A375, you see that they line up quite well along the diagonal. So again, each point here is a phosphosite, the value that we recorded for that phosphosite. Quite a bit of agreement between the two replicates. But if you compare a replicate of A375 Vemurafenib to Vemurafenib in PC3, correlation's low regardless of which metric you choose. So this is our first step at comparing profiles. We compute Spearman correlation between all of the profiles that we created in a single cell type. So then we get our next matrices, which are similarity matrices, but it could also be correlation matrices. We call them similarities because let's say we decided to swap out correlation for something else. It's still a similarity metric, so we call it the similarity matrices. And these are the samples of Vemurafenib. So again, the 6 cell lines by the 6 cell lines. The diagonal is 1, meaning each replicate looks exactly like itself. The other thing that is important to find is these clumps. So those clumps are the 3 replicates in the same cell line. So what you want to see is those clumps mean that replicates are consistent, which is something we definitely want in our data to be reproducible. And from a quick glance, it's clear that A375 kind of looks different, but we think we can do a little bit better. There's kind of 2 problems with this. I'll touch on in 2 slides. So this is the similarity matrix. I guess one other thing to point out is you can see some subtleties like NPCs here. Two of the profiles here correlate really well, but one of them doesn't. So now we've got some evidence that maybe we shouldn't use that replicate. And another kind of important note here is that, it's clear with Vemurafenib, but with any drug, the cell line in which the treatment was performed makes a big difference. So we can almost, if you sort of, regardless of treatment, cell line makes a really big difference when we just randomly cluster our data. Okay, so this idea of hierarchical clustering we've thrown around a lot of times, very familiar concept, but again, I wanted to kind of slow down and go into its guts a little bit. Clustering is based on the similarities that we computed on the previous slide, right? We computed all the pairwise similarities, using Spearman or whatever other metric we wanted to, but then we can rearrange our samples using those similarities so that we can visualize some sort of structure in the data. So these 2 samples, for example, get clumped together because they have the highest correlation out of any other samples. Hierarchical clustering basically goes 1 link at a time. It says, show me the 2 samples that are most similar and group those together. Then show me the next 2 that are most similar, group those together. Hierarchical, because then you build up to include all the samples. There's some subtleties about which linkage methods you can choose. So average says, after you've grouped these 2 profiles together, you need to somehow figure out what cluster is this most similar to. So you can either choose the very worst correlation of, basically each of these samples has correlations with all the other samples. So you can either choose this one or this one. So average says just choose the average of those. Single says choose the best. Complete says choose the worst. Kind of a detail. Right, but hierarchical clustering, also very familiar, allows us to sort of structure our data. But same with the similarity matrices, 2 things that we want to do a little bit better is 1, get rid of the replicates, and 2 is we want to have some sense of whether a particular correlation is significant. So dealing with replicates, there's different ways to do it. One way is you could just merge the replicates somehow. You just kind of average together the signal into a single signature. We choose to do it a slightly different way. We keep the replicates around for as long as we can. And I'm going to show you kind of big picture how we think about it. So we started with profiles, computed correlations, and then we end up with connectivity. And this step here, I'm going to explain in the next slides exactly what we do there, but the big picture, again, is to collapse replicates and just be able to say how similar is a compound to some other compound. And we call them perturbations or perturbations rather than compounds because it's not only drugs that we can do this with. We have CRISPR and short hairpins, so we call them perturbations. Okay, so how do we compute the connectivities? Well, we start with the Spearman correlations, the similarity matrix. It looks like this. This is toy data. We've got 3 drugs, A, B, and C, in the rows and the columns. So for every pair of compounds, we do this procedure. So let's take drug A and drug B. We'll call the first one the query. It's in the columns. And the second one the target. It's in the rows. And first we pull out all of the correlations corresponding to those 2 drugs, the replicates of those 2 drugs against each other. So that's 9 values. But then the comparison to a background, the second motivation, is for the target, all the other correlations. Except we exclude this little area because it's double counting, and we exclude these because those are self-similarities, so nothing special there. All right? So we have 2 distributions of numbers. We need to compare our 2 distributions somehow. We compare them with something called, well, we have to compare them. They look like that. We compare them with the Kolmogorov-Smirnov two-sample test mentioned briefly earlier in Jake's evaluation script. That's the same KS test. And what it does is it plots the two distributions on a cumulative probability plot. So what that says is, the way to read one of these plots is 1 is 100%, 0 is none of them. So at a given value, for example, 0, how many of the points are less than that value? So for the blue line, 30% of the points are less than 0. For the red line, it's something like 45% of the points are less than 0. And once you convert it to these cumulative probability plots, the maximum distance between those two lines is what the KS test spits out as its final statistic. If the distributions are really different, these two lines will be very far apart. A lot of the points will be, to the left or to the right. So we use the KS test as a proxy for how far apart the two distributions are. If they're really far apart, that's a high value, and we consider that to be significant. So this goes from 0 to 1, right? If the red line was all the way up the top and the blue line was in the bottom, the maximum distance would be 1. If they were exactly the same, it would be 0. But then we add a sign to it because we think there's something different about whether, if the test distribution values, if the correlations are less than the average correlation for a compound, we think that's different than if the correlations are higher than the average correlation for a compound. So we sign it based on which way it goes. So if the test distribution median is to the right, it stays positive, but if it's to the left, it gets a negative sign. Does that make sense? So all this is kind of the nitty-gritty of how we compute this thing that we call the connectivity score. It goes from -1 to 1, just like correlations. This is how it looks for real data. So if we have two compounds, belinostat and DMSO, the background is all the correlations for DMSO here. The test is the correlations between DMSO and belinostat. And the connectivity score is 0.42, which is not a very big value. And it's negative because the test distribution is to the left. Belinostat versus vorinostat is a high connectivity score. The distributions are very far apart. It means that the correlations between belinostat and vorinostat are much higher than the typical correlations for vorinostat. And it's positively signed because the test distribution is to the right. And then just like for the similarity matrix, we have a connectivity matrix, but it's smaller because we've gotten rid of the replicates. Now it's very clear that A375 looks different. It actually negatively connects with some of the other cell lines, and it's some ideas of how to explain the negative connectivity, but the main idea here is that it's clear that it's different. This is our framework. Yeah, a negative connection and a positive connection are different. And what it means is that the correlations underlying the connectivity score, they were smaller than the typical correlations to one of the compounds. So it doesn't mean the correlations were negative, it just means that they were smaller than the typical correlations.
The other compounds.
Yeah. Yeah.
Can you go back one slide? So here for the belinostat and vorinostat, you have the background is the typical correlations for the belinostat.
Yeah.
But if you compare it the otherwise, is then the background for the other?
No, yes, no, you're right. It's the second compound, the target compound that we use as the background. So the reason that's why it changes is because the second compound is different.
Okay.
That's right. So that's a subtlety, is that connectivity scores, the way we compute them, are asymmetric. Depends on which order you're doing them. We think that's intentional to be able to say some compound might be the closest one to some other compound, but there may be, it's a little hard to say in the air, but basically a certain compound might be the closest one to some other compound, but it could be in a tight cluster itself, so its closest neighbor is some other drug. Why is it a negative connectivity here? Yeah. So the test distribution is the correlations between belinostat and DMSO. There's some value. They're all these values, right? So but typically the correlations to DMSO are the blue values. Since the green plot is to the left of the blue plot, we assign it a negative connectivity score. These particular correlations are lower than the typical correlation to DMSO. So once we have these connectivities, we've got more matrices to play with. And a quick summary of why we do this extra step rather than just correlations. It's the way we collapse the replicates, and it's the way in which we compare the similarities to some background. And we chose a compound-specific background rather than the global background. But there's a bonus, which is that once you're in the space of connectivities, you can compare to other assays. So Jake's already mentioned earlier this GCP assay. Measures 59 changes to global chromatin. And we produced data for GCP on exactly the same samples as the P100. But we couldn't directly compare them because they measure totally different things. But once you have connectivities, you can say drug A and drug B have a high connectivity in GCP, but they have a low connectivity in P100. So perhaps those two drugs act on chromatin and not on the phosphoproteome, which is why they look similar here, but not similar there. That's kind of one of the ways you can use connectivity. And the other assay that we've been comparing to is L1000, which is transcriptomic. And again, they measure totally different stuff, but through this connectivity lens, asking, does this compound look like some other compound? That's how we can compare assays. We can do one more thing with the connectivities, which is we've been thinking about how to visualize them. So these heat maps are fine, but we get tired of looking at heat maps at some point. So we have these network visualizations. So coming back to our friend Vemurafenib, here is shown as a node. Each of the nodes here is a compound. And what we're doing here is we're plotting the connectivity scores, but we're showing only the scores above a certain threshold. So for example, we've chose some threshold, and now these lines here are the connections to Vemurafenib that met that threshold. But the other thing that we show is among those first order connections, any connections among those that also met the threshold. So you see that Vemurafenib connects to these two compounds, and those two compounds also connect to each other. That gives us more confidence that it makes sense, right? It connects to something, those things connect to each other. And similarly, Vemurafenib connects negatively to this compound C646, which connects negatively to the things that Vemurafenib was positively connected to. So all this is consistent. And then you've got some other connections to Spearman sort of far out by itself. But it allows you to zoom in on some compound of interest and get a quick glance of what's its neighborhood, what are the compounds it connects to, and are those compounds consistent with each other. There's a subtlety here, right? I mentioned that our connectivities are asymmetric, but here we have one line between 2 points. So we could have two lines, and we do sometimes have both lines with direction shown. That's a directed graph. But we've often found that symmetrizing for these graphs just kind of makes it easier to look at. All right, so that's kind of the computational framework. And going forward, I had 5 short stories of how we use our data to learn something about biology. Yeah?
Connectivity, you mean that you're also including the non-specific effects of each drug and then showing that these are the non-specific effects, that's why they are connected?
Yeah, that's true. We don't know if the connection between some compounds is because of their on-target or off-target effects. We just know that their profiles look similar.
But the non-specific effects can be really highly overlapped in drugs.
Right. Right. So the way we get at that partially is that the very first slide, those profiles were median normalized, right? So across the whole plate, we subtract out the median profile. So if most drugs all hit one probe up, it would look, that probe would be just blank across the whole plate because every drug hit that probe. So that's where the row median normalization helps. Anything else? Okay.
So you're collapsing the tickets by average, that's the first step before connecting.
We don't do that. That's one thing we could have done. We could have just collapsed the profiles and just had one profile for each signature, and then you don't have to deal with them. But we keep them around. We basically collapse them at the last possible moment where we compare the distributions of correlations. So if we only had, let's say we had 2 drugs with 3 replicates each, we could have collapsed them both into a single signature and compared those two, we'd get 1 value back. Instead, we keep 3 replicates around and do the 3 by 3, and we have 9 numbers that we can use to compare against some other background. So we think that keeping those 9 numbers around gives us a little bit more power than if we just collapse them right away into a cleaner signature probably, but then you only get 1 number for each pairwise comparison.
So you're comparing 9 versus 9.
We compare 9 versus actually many more, yes, because we have a bigger background. That's right. Okay, so first story. If we look at, what if we look at compounds annotated as doing the same thing? So if their annotated mechanism of action is the same, do they look like each other? We have, in our 90 compounds, we have 22 classes that have at least 2 members. And for each of those classes, we said, compute the connectivity between those members, and then aggregate them together. And then we also aggregated across cell lines here, because remember, we also had 6 cell models. So what this plot tells you is that BRD inhibitors, there's 3 of them, their connectivity typically is very high, 0.9, in every cell model, because the median here is 0.9. Well, not in every, but in aggregate. So there's a bunch of classes with positive connectivity, but there's at least one with negative connectivity or just low connectivities. So what if we dig into those? So BRD is kind of the good, right? Three compounds annotated as doing the same thing, all connect to each other. It's pretty cool. Then we have this class SIR2 and active, which is sort of in the middle of the pack. But there's kind of this lighter element here. So one of the drugs, it looks like, might be doing something a little bit different than the other 2. And this plot doesn't show it as clearly, but so we grouped all these 3 compounds together and called it SIR2 and active, but actually resveratrol typically is annotated as an activator. So it's got kind of ambiguous annotation generally in the literature, but typically it's an activator. So you can see there's discordance to some degree between the inhibitors and the activators, but also some kind of commonality in the signaling pathways that they're targeting. So this is an aside, but if you look in GCP, the assay that monitors chromatin, SIR2ins are chromatin related. In GCP, you can see clear as day that the activator and the inhibitor are totally different. And I didn't have it here, but the fact that we can make that comparison is because of connectivity, right? We're talking about this compound looking like some other compound, but in totally different assays. We're measuring totally different things. Last one is the junk inhibitor, which there's 2 junk inhibitors, and the kind of white here is to show that they just don't have connectivity. So that tells us that those compounds, even though they're annotated as doing the same thing, either are misannotated or there's significant off-target effects in one or both that are making their profiles look different. So that's one way. That was kind of a global view of what you can learn from the connectivity. Another one is coming back to our old friend Vemurafenib. So this story is very similar to the one that I've been covering so far, but it's a little bit more subtle. So we've already seen that Vemurafenib should do something different in A375 than in other cell lines, right? Not only that, you can also see that its connectivity to 2 other compounds, these MEK inhibitors, is only very high in that one cell line. And the reason for that is that Vemurafenib hitting BRAF here looks very similar to hitting MEK, which is immediately downstream, but only in A375 because that's the only cell line that has this target at all. So it's not just that Vemurafenib does something different, but the thing that it's doing different is looks a lot like MEK, which makes sense since it's immediately upstream.
One of the things to note is, and I'm not sure if you're going to answer this, Vemurafenib activates wild-type BRAF.
Yeah.
When there's no BRAF around.
That's right. So there's this paradoxical activation where Vemurafenib actually does other stuff that we don't want it to be doing, like activating BRAF, and so that's a problem in the clinic as well. So yeah, I mean, there's other stuff going on, right? But it's clear as day that these two compounds in the on-target cell model, there's something consistent going on. Third example is this compound Pazopanib. So Pazopanib is a receptor tyrosine kinase inhibitor, a receptor growth factor receptor inhibitor. It's kind of a sloppy compound, but what we want to show here is if we look at what this compound connects to in each of the cell lines. So here we're showing we're sorting the matrix by the median across the row. So the thing that it connects to most is itself. That's good. That means there's consistency in its replicates. But then the things that are next in the list are negative connections in only one cell model, in the NPCs, which are neural progenitor cells. So in one of the models, this drug is doing something very different than it's doing in the other models. And even in more what's even more striking is that you then look specifically at NPCs and sort again, this is the top of the list and the bottom of the list, those compounds that were some of the very strongest connections in the other cell models are now the strongest negative connections. So we've dug into this a little bit, we don't have a great explanation, but it does kind of capture the fact that we're seeing some subtle cell-specific effects with this assay. And in particular in a model that it wasn't suited for. These are normal neural progenitor cells, not the cancer cell models that we've been doing all our work in. So example 4 is these network views that I talked about. So I showed some heat maps, but the network views are useful for seeing these neighborhoods. So what I've done here is I chose a very strict threshold. I said show only the top 0.5% of connections in the whole assay, and again aggregate across cell lines, and then just display what we've got. So there's some things that are consistent and nice to see. These 3 compounds here are all BRD inhibitors, and they have strong correlation, strong connectivities. These are the 2 MEK inhibitors that we saw earlier, and what's interesting is you don't see the connection to Vemurafenib because typically in the other cell models that's not a strong connection. You see 2 of the HDAC inhibitors, but there's other HDAC inhibitors that we profiled, so it sort of says the very strongest connection that remained was between these 2. This other one has these connections to other compounds. There's this other kind of unexplained connection. And then there's this kind of big clump of compounds which include the cell cycle inhibitors. So those drugs that are annotated as doing other stuff, like acetyltransferase inhibitor or a ubiquitin ligase inhibitor, they're involved somehow with cell cycle, we believe. And that you can see by this neighborhood showing up even in the very strongest connections. So the last story is not P100, but it uses this connectivity that I've spent a lot of time explaining. So here, quick recap of what this GCP is. So again, these are your histones with DNA wrapped around it. The histones have these tails that can get modified, and that affects what gets transcribed and what doesn't. And we can measure very specific combinations of those modifications. So we can measure histone 3, lysine 4 unmethylated when it's monomethylated, dimethylated, acetylated combinations of ubiquitination and acetylation and methylation, and we have 59 of these. So in the same way we did with P100, we profile a bunch of cells treated with drugs, and we get a little matrix like this, big matrix, 59 rows for the 59 features we measured, and 540 perturbations for 90 compounds by 6 cell lines. But instead of just looking at this matrix, we also made more data. This box, which is again GCP, 59 features, but then we profiled 804 cancer cell lines, not under perturbation. So just at baseline. And then we said, can we take these vectors and compare them to these vectors using that same framework of connectivity? And we get a matrix like this, where we've got our 540 perturbations by 804 cell lines. And now we can do a cool thing and ask, are there any cell lines whose baseline state looks like inhibition with a drug. So as an example, we'll look at EZH2. So EZH2 is a lysine methyltransferase. And we have 5 cell lines that have a mutation in EZH2. These are all point mutants. And if you ask, what are the things, what are the perturbations that most connect to those mutants, the top 18 results are EZH2 inhibitors. So that's a perfect result. And in fact, if you kind of look at the whole picture, the whole list, this is 540, all the EZH2s up there. So it's not even a pretty picture because all the lines are too close together at the top. But this is kind of a good, this is a really positive result for us. There's a more nuanced example that's very similar, looking now at NSD2 mutants. So NSD2 is a nuclear receptor binding domain protein. Don't know much about NSD2, but there are 2 types of NSD2 mutants that we have in our data set. So one of them is caused by a translocation that messes with that protein, with that gene, and the other one is a point mutant. So you can see that they clustered, the 2 types of features clustered to themselves. But then if you now look at what are the connections of those classes of compounds to, those cell lines to the compounds, and now we're looking at the bottom of the list, we see some interesting things. So this set of cell lines here, these are all multiple myeloma cells, and they're enriched for negative connections for HDAC inhibitors. So the hypothesis was, if we have a drug that looks the opposite of the natural state of a cell, perhaps that drug could be used as a therapeutic. It could undo whatever mutations in that cell line we're doing. So we looked in the literature, and indeed there's HDAC inhibitors being proposed for multiple myeloma. And on the other side, there's other classes of compounds that are enriched for negative connections to this set of cell lines, and I've only shown some mTOR references here, but again, there's evidence that those compounds that look the opposite of the baseline state of a cell could be used as a therapeutic. So, those are my 5 stories. What do you think? Any questions so far?
Sorry to keep harping on that 80% but like, do you have other melanoma lines in there?
No.
So I know it's used a lot in the literature as a model.
Yeah.
But the thing I always found weird about it is a homozygous BRAF mutant.
Okay.
And so I don't know if you sort of vetted the other lines to see if there's canonical driver mutations associated with them.
Right.
Are you tended, the cells you chose to derive this, they tend to be sort of like 80% homozygous driver mutations, or do you have a lot more heterozygous driven cases
Yeah.
in there?
I don't know if we have that level of characterization. I think they were chosen to represent a diversity of cancer lines, and then we threw in one, or a diversity of lineages, and then we threw in a totally non-cancer line to then also increase our space that way. Yeah. But it's true. I mean, we could go deeper in the space of cell lines, but there's so many other ways that we could go deep too, like dose, compound, genetic perturbation. So it's kind of a game of figuring out exactly what question to pursue.
Right, I'm just wondering sort of what your space looks like
Yeah.
currently, rather than
Yeah.
So I presume then, like, if you just run any A cell line, say a resistant cell line, you can predict the drug response based on if you're using the same kind of assay.
Yeah.
Right?
Could you say that again?
Like, say now you're running cell line Z.
Yes.
Right?
Okay.
A resistant cell line to some drug.
Okay. Yeah.
Now, from your data, if you're running the same assay, then you can predict which other drugs will be responsive. to
That's
that
right.
profile that
That's
you're
right.
getting from that cell line.
That's right. You could profile a new cell line and say, do we have any signatures in our database that look the opposite of this cell line's baseline expression? That's one approach. We kind of, I think this one is a little bit more robust in that we use a whole group of cell lines. So maybe the aggregating across a few cell lines might help with that. But yeah, the idea is extendable.
So is there any easy way of doing that in your database or whatever?
Yeah, we're in the process of building an app that would allow you to bring your own data to the table and query our resource. Because right now, in fact, in the tutorial, we'll play around with the data that's publicly available. But yeah, we want to also enable people to bring their own data and query our resource. So it's in the works. You have to have some overlap with our features. Yeah.
Or good overlap.
Yeah.
Regarding the example GCP features from it.
Mm-hmm.
So these genetic marks are basically they have a specific meaning, right? So it could be one regressive or it activates something. So
Right.
did you find any correlation between these GCP features with your compounds and your cell lines?
Right. So if you know, could we learn that a certain compound hits a very particular site just by looking at what happened to that site when we treated with that drug? I think we've, for the most part, avoided looking at individual features because of the very first slide of how you could look at individual features and try to learn something that way. I guess with GCP it might be a little different, but with P100 especially, we just know very little about the sites themselves.
Because some of these features are really very opposing features. So
Mm-hmm. Yes. Yeah, so we'll see, for example, if lack of methylation goes down, these other two will go up. So the features are not independent the way we have them. Yeah. Anything else? All right, so should we do a little tutorial of Morpheus then?
Is the code to generate connectivity course available?
Yes, we have a GitHub page we can point you to it. All right, so Morpheus can be found in two places. We've been advertising the first link, but been finding it's a little slow, so if that's not working for you, you can try the second link. But why don't we all open up Morpheus now, and we can walk through an example together. All right, so we have Morpheus, and then the file that we'll analyze is also on Panorama. So if you go back to Jake's page, we store our similarity and connectivity matrices under this heading here. If you click on that, you can see the similarity and connectivity matrices for the different cell lines in both assays. So why don't we stick with P100, and we can choose why don't we do P100 A375. And if you just click on it there, actually I guess we didn't even need to open Morpheus, it'll just take you to Morpheus. So, is everyone there? Do we have the matrices open? If you go to panoramaweb.org/labkey/project, you can also search links panorama. And when you're on the site, if you click on links, then similarity and connectivity matrices should be in the drop-down menu. So does everyone have that matrix open? Okay. And then similarity and connectivity matrices. Ah, yes, and then one of the connectivity matrices, so if you want to be on the same page, I chose P100 A375 here. Near the bottom. Connectivity matrices. below similarity, there's connectivity. So similarity is just the Spearman correlations that I had mentioned first. So are we all there? Okay. So when you open it up, the default is to color each row by the minimum and the maximum. So I like to undo that to get a sense of the global picture. So if you click on the gear here, guess it doesn't like me expanding. All right, can everyone see the gear here under options? And then for color scheme, you can either uncheck this so it's no longer relative and then set the minimum to -1. Yeah. And then the other thing is the annotation. So it spits out a few annotations. We can throw out from the row annotations, we can remove cell ID because we know it's all A375. So you can unclick it. And then in columns, let's also unclick cell ID. Okay. We have our matrix here. Let's say we wanted to look at that Vemurafenib and MEK connection. So that requires a search. If you go to the, let's start with the rows. Let's search in the rows for MEK. So it suggests you might want the MOA, the mechanism of action, that is MEK. It tells you there's 2 matches. But how about in the columns, we want a specific compound, that's Vemurafenib. And it suggests PERD-I name, perturbagen name is Vemurafenib. So you can select that. So now it's 2 rows and 1 column selected. Does everybody have that?
Say MEK,
M-E-K.
Oh. And I'm sorry, we have both PERD-I name and MOA in both the top and the bottom?
No, in the rows, we'll search for just MEK, and it'll suggest the MOA, MEK inhibitor. And then click on columns to now search for the PERD-I name, Vemurafenib. Let me see if I can make this a little bit. How are we doing?
I'm getting the wrong direction though.
The wrong selection?
Yes, some BRAF inhibitor is getting selected. Calpain inhibitor.
Hmm.
Why is that? Shows MEK inhibitor.
You want to make sure that the only thing in that search box, that there's nothing else also?
No.
Hmm.
It says 2 rows, 1 column selected, but then I see a blue on BRAF inhibitor.
Ah, that's good. That's the Vemurafenib. Vemurafenib's the RAF,
Oh, yeah,
the RAF
yeah,
inhibitor.
that's fine. But
2
here-
rows and 1 column.
Oh, and the rows?
And the rows are down lower in here. They're the MEK inhibitors. So 2 rows, 1 column mostly we've got. And if you want to just look at those, you can press this up arrow to send the matches to the top. Doesn't help that much. Let's say you wanted to just isolate those 2 to their own little heat map, like the ones I'm showing in the slide deck. So you can just basically cut out that selection and make it its own heat map by doing. So on a Mac, the shortcut is just Command X, but in case you need the full thing, we want to do new heat map. And the new heat map is just going to return the things that we had selected. Any luck?
Can you just walk us through
One more time. Yep. So
I only have one thing selected, but I see you have multiple.
Yes.
You just scroll down.
yes,
Okay.
and it says up here what you have selected.
Yes.
So if you have that, you can make it its own heat map by doing Command X or by from the drop down Tools New Heat Map. And you can see it opens up a new tab with just your selection. So that's kind of the basic stuff of just isolating the data that you want to look at. But it does all sorts of stuff. So let's say we want to try clustering, famous hierarchical clustering. So that's also under Tools. If you click on Tools and then Hierarchical Clustering. So now I've gone back to the original heat map. So I just clicked on the other tab. If you click on Hierarchical Clustering, it gives you a bunch of options, some of which we've discussed, right? So the metric is each of the columns or the rows. How do you want to say how far apart they are? You can either use Pearson correlation, Spearman correlation, Euclidean distance, or a whole bunch of others. So let's say we wanted to stick with Spearman. Select Spearman. Linkage method, we can keep average and cluster just the columns. Is everybody here attempting a cluster? Okay. And then, okay. And you get your dendrogram back. Okay, pretty cool. Some structure in the data. Let's say we wanted to do a similar analysis to the very first one I showed, which is to say, are compounds with the same MOA, are they similar to each other? So we can kind of look by eye here and see that there might be some MOAs that are similar, like these HDAC inhibitors here. But we can also add a color to the MOA to make it even clearer. So if you right-click on MOA and select Show Color, then you get a little color box next to each label. So now maybe it's a little easier to see you've got some HDAC inhibitors, some cell cycle inhibitors, these BRD inhibitors that we had pointed out. And let's say we wanted to zoom all the way out and see the whole picture. We would go to View and click Fit to Window. So that would shrink it all until it fits in our window. And you can get a really far out view where it shrunk all the rows but kept the columns the same about which compounds look like which other compounds. So this is kind of a subtlety. We're now clustering the connectivity matrix. So we're looking at the connectivities to see whether compounds look the same based on the things that they connect to are compounds similar to each other. Everyone get a basic flavor of hierarchical clustering? If you remember, I showed the scatter plot briefly on real data. So that was also with Morpheus. If you select more than one column, let's say, so I selected this RAF and this MEK inhibitor. And now in the rows, if I click on a row, and then I can just say drag and select all the rows like that, or do Command A or Control A to select all the rows. Then when you go to Tools and Chart, Tools to Chart, it'll give you a bunch of options on what kind of chart you want to make. So let's say I want to just scatter those connectivities now for the two columns I selected. So I could do a column scatter. And in the far right, it'll show me the scatter plot of the two columns that I had selected. So I know this might be really, really fast, but I'm trying to give a little bit of a taste of kind of the things that are possible. And I guess the main thing is that what Morpheus consumes, what it consumed in this case was this GCT file that Jake has mentioned, but it will work with regular old tables with labels, CSVs. So you can bring your data to it and it'll display it and you can interact with it. And it's just, I use this tool more than anything else, and it's just, you know, not for lack of other tools, but it's just so intuitive and so easy to get started. Yeah, you can save the image as a PNG or a SVG, you can save the whole data set. You can save the whole session like Jake did, so you can share a view with a collaborator. All right, so I think that's kind of a flavor. Unless there's any specific questions on what you can and can't do, maybe we could wrap it up here. Anything else?
Just the data, if you want to use your own data,
Yep.
because it has to be some format like the headers or the column names or.
I think it'll just eat a regular CSV. It'll take an Excel spreadsheet.
But you can also make your own GCT.
Yep, and there's a whole suite of tools that we have for making GCTs from R objects and Python objects and saving and manipulating them.
Is there a GCT repository somewhere, or just?
It's called L1K Tools, is the GitHub repository. It's actually soon being deprecated and split up into different language-specific ones, but for now, L1K Tools is the place to go.
No, I mean,
And that-
repository of GCTs made from other projects.
No. Jake's website is the main source of free GCTs you can find, I guess.